Hello everybody and welcome to Mr. Saucedo's YouTube videos. Today we're going to be going over the first lesson all about DNA and how it was discovered. So just to kind of give you some background information though, right? So we're going to kind of stop midway through the lesson, but this is all about how DNA is structured, and then we're going to go over some scientists that kind of helped to discover what DNA does, and then in the next lesson, kind of more about like how it's structured. So first things first, in our unit 1 that we did all the way back in like August, we were talking about how there are characteristics that all living things share, and one of those was that all living things have a universal genetic code, which is made up of DNA. So the very first kind of guy that we're going to be talking about is Frederick Griffith, and so that's a picture of him looking kind of mad for some reason. And so we've got these categories. So he was doing an experiment where he was injecting mice with bacteria and wanting to see kind of what happens as a result. And so what we're going to do is kind of fill in this chart as we go. You don't need to color code it or anything. I just colored it in a little bit just to make it look a little bit less plain. So our very first group, that particular group, lab group 1, was injected with like a low risk type of bacteria. So, you know, something that's not really going to be fatal, not really going to cause death. And that bacteria was completely alive. So that mouse was injected with a bacteria that was alive. And so what we're going to do is we're going to basically put X's over things that we think die. What do you think happened to that particular mouse in that lab group? Spoiler alert, it lives, right? Because it's a low-risk bacteria. In the second one, though, he injected this particular mouse with a high-risk bacteria. And that bacteria is also alive. So what do you think happens to that mouse in that situation? Answer, it dies, which again is not unexpected, right? Like this is a lot more fatal. It's high risk. It's alive. The mouse is probably not going to do too well. In the third group, though, he took the high risk one again, and he essentially like killed the bacteria with heat. So he kind of like heated it up so the bacteria was dead. So he injected this mouse with dead bacteria, but it's the high risk kind. So what do you think happens to that mouse? Answer, it lives. And again, not really unexpected, right? But for the fourth experiment, here's where it gets interesting. He decides to inject the mouse with a mixture of the high risk and the low risk bacteria. So for the high risk one, he kills it, just like he did for lab group 3. And for the low risk one, he leaves it alive. So essentially, he's mixing lab group 1 and lab group 3 together and injecting those bacteria into the mouse. What do you think happens to the mouse in that situation? See, the mouse dies. And that is super interesting because you wouldn't expect that, right? Lab group 1, it's low risk. It's alive. It lived. Lab group 3, it's high risk. And it's dead. So you're essentially injecting, something that's dead into the mouse and it lives. Why, when you put them together, is the mouse dying? All right? And so what he basically said was that bacteria, even in death, even if the bacteria is dead, could transfer some genetic information to living bacteria. And again, genetic information is in quotes here because he wasn't sure what it was. And so just to give you an idea, here's like, you can freeze it and kind of like, pause the video if you need to. Here's a general kind of description in comic form of kind of what he did and his conclusion. All right, now we're going to fast forward over a decade later. We've got this guy named Oswald Avery, and he's doing something very similar, but he wants to modify it. He wants to change the experiment a little bit because what he's interested in is what is that genetic information, right? It could be anything. And so the 3 categories of molecule that he thought it could be were proteins. We'll talk about the ACE part, RNA, or DNA. Those are the 3 categories that he was thinking could be what's carrying genetic information. So remember, enzymes we talked about all the way back in first semester, they end in ase typically, and it tells you kind of the beginning part of the word what it does. So protease destroys protein. So in the bacteria that is being injected into these mice, there's going to be no protein being injected. In the second one, there's going to be no RNA being injected. In the third one, there's going to be no DNA being injected. And I know we haven't really talked about RNA, but we will in this unit at some point. But just know that's one of the other categories of molecule out there. So what type of bacteria again? Well, he wanted to do the exact same mixture from Frederick Griffith's experiment. So high risk and low risk mixed together. Are they alive or dead? He wanted it to match exactly. The high risk one is dead, the low risk one is alive. So what do you think happens to the mouse when the bacteria is injected, but all of the protein has been removed from the virus, from the bacteria? The mouse died. Still, just like Frederick Griffith's experiment. What about when you remove all of the RNA from the bacteria? The mouse died. When you removed all of the DNA, though, the mouse lived. And so what he concluded here, and this isn't actually his conclusion, but he knew that that genetic information was likely going to be DNA. Nobody really knew entirely what DNA was used for when Frederick Griffith was doing his experiments in the 1920s. Even by the 40s here, there were different ideas. So there were biologists that kind of thought proteins or RNA or DNA were storing genetic information, or it was a mixture of all of them. And so again, just if you want to pause it, you can take a look at our experiment here, right, for Oswald Avery. Okay. Big conclusion, what is DNA used for then? He was thinking DNA must be used to store and transmit genetic information. And so essentially it was a baton pass, so that dead bacteria was able to still pass on its DNA to the living bacteria. And so you can think of it as just kind of passing on the torch to the one that's alive. And that was interesting. Now, again, even years later, scientists aren't entirely sure whether DNA is the only thing that transmits genetic information and stores genetic information. So these two scientists, Alfred Hershey and Martha Chase, they decide in the 50s to prove that DNA is the only thing that carries our genetic information. Here's a nice little picture of them. And the way that they're going to do it is not with mice. They're going to use viruses. So there are viruses, they're called bacteriophages. They're the most populous kind of virus on Earth, and they only infect bacteria. So what they did was they decided to use radioactive dyes to see what is the virus injecting into the bacteria, right? Is it protein? Is it DNA? Is it DNA and protein? And so here's a picture of like a bacteriophage. You can kind of see them here. This glowing bit that you're looking at here that's kind of like bright white, that would be like a radioactive dye. And so this little stringy thing that you see here, that's viral DNA. And then this yellow stuff here, that is the bacteria. Okay. What did they find? They found that DNA is what is being injected by these viruses. Not protein and DNA, only DNA. And so they concluded that DNA is the only substance that carries our genetic information. Okay, so just to kind of review some stuff that you probably already know. How is DNA structured? Well, first of all, DNA stands for deoxyribonucleic acid. That's the D, N, and the A. And so DNA is just made up of a bunch of nucleotides. Nucleotides are joined together by these covalent bonds, and that's kind of what makes our DNA. So what exactly is a nucleotide? This is a diagram of a nucleotide. So make sure you draw it in your notes, you color code it, and you label it. So the phosphate is this orange bit here that kind of looks like a circle. We have a 5-carbon sugar. That's this little pentagon in blue. And then you've got this green nitrogenous base attached to it. This is what a nucleotide is. And what is this 5-carbon sugar? It's called deoxyribose. That's where the beginning of DNA gets its name from, deoxyribo. And then nucleic acid, it is in the nucleus. Okay. Last but not least, this is the end. There are 4 types of nitrogenous bases in DNA. You probably are already aware of that. You've got your A, your T, your C, and your G. A stands for adenine, T stands for thymine, C stands for cytosine, and then G stands for guanine. All right, if you have any questions, please let me know. Make sure you fill that out in your notes.